relative Drosophila simulans is native to Madagascar. Simulans is known for spawning a couple of closely related island endemics, one of which, Drosophila sechellia, from the Seychelles, not surprisingly, is famous for being adapted to a toxic fruit. That is the topic of this talk. But I'm also going to say a lot about Drosophila yakuba in this talk. Yakuba is primarily known as a mainland generalist species, native to savannas across western, central, and eastern Africa. But, and there are several other species as well, which I don't have time to include in this background. But what I'll be talking about today is kind of a special population of Drosophila yakuba that found itself on an island between Madagascar and the mainland called Mayotte, and what happened next. So on Mayotte, instead of behaving like a normal generalist population of Drosophila yakuba, it was only found around noni fruit trees. And so this is a curious species, the noni that is. It's able to disperse across great distances of seawater because its seeds can remain viable for weeks in that context. And so, not surprisingly, you tend to find these trees often growing around coastal areas. And natively, it's distributed throughout the Indian Ocean and Oceania, like the Western Pacific, although it's also colonized other areas like the Caribbean in more recent times. On Mayotte specifically, there were no humans in this part of the world, including Madagascar, until within the past 1,500 years. And so, the array of fruits that humans tend to bring with them weren't there originally. And so, one notable thing about noni is that it does fruit year-round, and so it may have, presented kind of a rare, predictable resource to Drosophila who are arriving on the island. The challenge, though, is that it's also toxic, and in particular, it contains octanoic acid, which is toxic to the great majority of fly species in this clade. Drosophila sechellia, though I mentioned, is a known case of noni specialism. What we found is that this Mayotte population of Drosophila yakuba, though, is also a noni specialist. And, again, it was found on and around these noni fruits and trees, but even though Mayotte is inhabited today, and there are some nice, mango orchards and things like that, yakuba doesn't seem to like them. So it's almost never found in those environments. And so when my colleagues actually tested the olfactory preferences, or the behavioral preferences of these flies, what they found in this smell test is that the mainland yakuba, if offered either banana or noni traps, will disproportionately choose banana. But the Mayotte flies will even more disproportionately choose noni over banana. So, and almost to the degree that sechellia does. And in terms of tolerance of noni fruit, this is using whole fruit for tolerance testing of adult flies. You can see that the majority of mainland flies, the red bar here, are killed by noni in this particular experimental context. But the great majority of Mayotte yakuba can survive that, and they survive almost to the degree that sechellia does. And in fact, the F1s are doing just about as good as sechellia here. And the F1s also point out that although there could be an element of hybrid vigor involved here, these F1s between island and mainland yakuba show that tolerance appears to be largely dominant, and that attraction appears to be, partially dominant, perhaps mostly dominant as well. So this is building the case, along with the distribution on the island, that yakuba from Mayotte is actually a noni specialist and has kind of recapitulated the evolution that we've seen in Drosophila sechellia. Another curious little detail that I'll mention here, although it's a bit disconnected from the rest of the talk, is that these island flies actually show a degree of mating preferences. So they can mate with mainland flies, and if they have no choice, they often will. But if you give them a choice, island females would greatly prefer to mate with island males over the mainland males. And so there is a degree of asymmetric partial prezygotic isolation here that could be interesting to study further. I won't say anything more about that today, but it provides another dimension for further studies of these flies. So instead, I'm going to focus on the noni specialism angle. And I mentioned Drosophila sechellia, which was already a great model system for the genetic basis of shifting to this specialism on a toxic fruit. And there have been a lot of great QTL mapping and gene expression and other studies on that species. One limitation, though, is that sechellia got there millions of generations ago. And for that reason, population genetics has not been as useful in detecting the targets of selection with regard to this transition. And so we wondered, in light of the fact that, our populations still basically look like Drosophila yakuba, they can still interbreed pretty well, could this be a younger instance of noni specialism? And also, how much might be shared in common genetically between noni specialism in sechellia and yakuba? So we collected some pooled genomic data from the Mayat strains, and we compared them to published genomes from yakuba that were sequenced from populations from the mainland, from Cameroon and Kenya. And the first thing we did was try to get a handle on how different they were between the island and the mainland. We were talking about an FST of roughly 0.15, and doing some demographic estimation to give us kind of a rough sense of the history that may have been involved here. The model suggested that there may have been a population split roughly 30,000 years ago, with kind of a mild bottleneck on the way into the island population, as you might expect, and with a possibility of ongoing migration, although I feel like that's still tentative. But basically, the good news for us is that these populations are not so distantly related, and that Mayat yakuba may have originated relatively recently, providing good hope for population genetic approaches. And so the method that we used for population genetics here starts with FST among these three pairs of populations. So we have three FST values. We convert them to Reynolds distances so that they scale more linearly with divergence time. And then you can calculate this three-population FST-based statistic called PBS, which is basically just aiming at estimating the length of the Mayat-specific branch over here. And then we went one step further, and it's just slightly more elaborated summary statistic here that we call population branch excess. The thing about PBS is that it's going to be large if there's been something special that happened in Mayat, but it's also going to be large if every population has a long branch. And we were interested in things that might be more specific to Mayat, and so we came up with the idea of basically taking PBS observed minus PBS expected, where the latter is based on basically how big is this distance between the other two populations at this locus, and also genome-wide in terms of the median value, at least on that chromosome arm, and what is the usual relationship between PBS and that distance. So that's the statistic that we're using, and that should be more specific to population-specific evolution. This is the result of the genome scan. Don't have time to go into great detail on it. But we did find a lot of genes and gene clusters with either known or very plausible roles in detoxification especially. And some of these genes have also been implicated either in sechellia or in other host-shift Drosophilids. This is one example of this big X-linked peak here kind of breaks in two. And there are genes in both of these clusters. There's ecdysone oxidase in this GMC cluster, and GCE is also involved in ecdysone signaling, which is a pathway in insects that's often targeted by plant compounds. We were interested to take a more general perspective, though, on whether the population genetic outliers for Mayat yakuba might overlap disproportionately with QTLs that have been mapped in Drosophila sechellia, to try to ask whether some of the same genes, or at least gene clusters, might have been involved in adaptation to noni. And so we tried this for 2 different classes of QTLs. And for attraction, there was no hint of a relationship, really. P-value was like 0.2. But for octanoic acid tolerance, we got a significant result, suggesting that at least some of the same genes may have been involved in the adaptation of octanoic acid tolerance in these 2 species. So to summarize what I've told you in this talk, we've documented the second known instance of noni specialism in Drosophila, and the recent origin of this population of yakuba allows us to use population genetics as a tool to help identify targets of selection. I talked about PBE as an FST-based statistic to try to focus in on population-specific frequency change, and our results suggesting that there may be a degree of genetic parallelism, especially involved in the tolerance of octanoic acid. So I'd like to thank especially my co-authors who contributed to this study. Amir Yassine is starting up a new lab at the Museum of Natural History in Paris. And I'd also like to thank collaborators who provided data or materials or help to us, and Lounsbury Foundation and NIH for funding. So thank you for your kind attention.